Stage 4 colon cancer used to be a death sentence. Now researchers are watching tumors shrink, sometimes disappear completely, using something your body already produces naturally. What changed wasn't a new drug. It was understanding how cancer cells feed themselves and what happens when you cut off that supply. I'm Dr. Marcus Ellington, a regenerative medicine specialist. For the past 15 years, I've studied how cells repair themselves and what makes them fail. The breakthrough in colon cancer isn't complicated technology. It's exploiting a fundamental weakness that every cancer cell shares. They're addicted to specific nutrients in a way healthy cells aren't. Remove those nutrients strategically, and cancer cells enter what researchers call metabolic crisis. They don't know how to adapt. Healthy cells do. Here's what most people miss about cancer. You probably have microscopic cancer cells forming in your colon right now. That's normal biology. Your body makes copying errors constantly when cells divide. Some of those errors create abnormal cells. The question isn't whether you'll develop cancer cells. You already have them. The question is whether those cells can grow large enough to become dangerous. That depends entirely on two things. Can your immune system find and eliminate them? And can they access enough fuel to multiply? Stage 4 means the cancer has spread beyond the colon to distant organs. 10 years ago, 5-year survival rates hovered around 13%. Patients were told to get their affairs in order. Now we're seeing something remarkable. At major cancer centers, 2-year survival rates for metastatic colon cancer have jumped to 60% with new combination therapies. Some patients with stage 4 disease are achieving complete remission. The tumor markers in their blood drop to undetectable levels. Scans show the lesions shrinking, sometimes vanishing entirely. The shift came from understanding cancer metabolism. In 1931, a scientist named Otto Warburg won the Nobel Prize for discovering that cancer cells consume glucose differently than normal cells. Even when oxygen is plentiful, cancer cells prefer breaking down sugar through an inefficient process called glycolysis. They're burning fuel fast and dirty, generating energy quickly but wastefully. This seemed like a curiosity for decades. Why would cells choose a less efficient energy pathway? Turns out it's not about efficiency, it's about speed and building blocks. Cancer cells aren't trying to maximize energy production. They're trying to divide as rapidly as possible. The glycolysis pathway generates the raw materials they need to construct new cells. They're prioritizing growth over efficiency. This creates a specific vulnerability. Recent research from multiple universities has confirmed something striking. When you reduce available glucose and certain amino acids, cancer cells don't just slow down. They experience what looks like cellular panic. The metabolic pathways they rely on completely shut down. Without those specific nutrients, they can't build the components they need for division. They enter a crisis state. Many of them die outright. The technical term is apoptosis, programmed cell death. The cell essentially self-destructs. But here's what makes this approach powerful. Healthy cells respond completely differently to the same nutritional restriction. When glucose drops, normal cells switch to burning fat for energy. They have metabolic flexibility. They can use different fuel sources. Cancer cells have largely lost that ability. They're locked into their glucose-dependent pathway. This difference creates what researchers call differential stress resistance. Fasting or severe carbohydrate restriction protects healthy cells while simultaneously targeting cancer cells. Studies using mouse models with colon cancer showed this clearly. Researchers fed one group a ketogenic diet, very high fat and extremely low carbohydrate. The diet triggers the liver to produce compounds called ketone bodies. Healthy cells can burn ketones efficiently. Cancer cells struggle with them. In the mice on ketogenic diets, tumor growth slowed dramatically. In some cases, tumors stopped growing entirely. When researchers stopped the diet, tumors started expanding again. When they resumed the diet, growth halted. The effect was reproducible and directly tied to the metabolic intervention. One specific ketone body called beta-hydroxybutyrate appears particularly important. It activates signaling pathways in colon cells that tell them to stop dividing. It's like a molecular brake pedal. Cancer cells, already metabolically stressed, can't override this signal the way healthy cells can. The compound essentially freezes tumor growth. Now, the human data is more complex than mouse studies. Humans have different metabolisms, longer lifespans, and cancer that's often more advanced when detected. But clinical trials are showing consistent patterns. Patients with colon cancer who adopt ketogenic diets or periodic fasting protocols show slower disease progression. Some show tumor shrinkage. The interventions work best when combined with standard treatments, not as replacements for them. The mechanism goes beyond just glucose restriction. Cancer cells are also addicted to an amino acid called glutamine. Your body uses glutamine for dozens of normal functions. but cancer cells consume it at rates 10 to 50 times higher than normal cells. They need it to fuel the metabolic cycle that replenishes their energy systems. When researchers restrict glutamine in laboratory settings, colon cancer cells stop dividing and begin dying within days. Combining glucose restriction with glutamine limitation produces an even stronger effect. Studies show that this dual metabolic stress essentially starves cancer cells from multiple directions simultaneously. They can't compensate by switching to alternative pathways because you've blocked both primary routes. The practical question becomes how you implement this strategically. You can't just stop eating protein and carbohydrates indefinitely. Your healthy cells need those nutrients too. The approach requires precision, specific timing windows, carefully calculated nutritional composition, understanding which foods contain hidden sources of glucose and glutamine that would undermine the intervention. But before we get to the exact protocol, you need to understand something critical about your gut bacteria. Recent research discovered that the microorganisms living in your colon directly influence whether these metabolic interventions work or fail. The wrong bacterial populations can actually feed cancer cells alternative nutrients that bypass your dietary restrictions. The right bacterial strains produce compounds that amplify the cancer-starving effect. This is where the science gets particularly interesting, because the foods that shift your gut bacteria toward cancer-protective strains are the same foods that create the metabolic environment cancer cells can't survive in. Your gut houses trillions of bacteria. Most people think of them as passengers. They're not. They're active participants in your metabolism, your immunity, and critically, whether cancer cells can find alternative fuel when you try to starve them. A study from the University of Pittsburgh changed how researchers think about this. They took mice with colon cancer and divided them into groups. One group received a ketogenic diet. Another received the same diet, but first had their gut bacteria wiped out with antibiotics. The results were stark. Mice with intact gut bacteria on the ketogenic diet showed significant tumor suppression. Mice with depleted bacteria on the identical diet showed minimal benefit. The diet alone wasn't enough. The bacterial ecosystem determined whether the metabolic intervention worked. The mechanism involves a fatty acid called stearic acid. When certain bacterial species are present and you consume high amounts of fat with minimal carbohydrates, those bacteria produce elevated levels of stearic acid in your colon. Stearic acid does something remarkable. It triggers programmed cell death specifically in cancer cells while leaving healthy colon cells unaffected. It's selective toxicity at the molecular level. Here's what makes this particularly powerful. Cancer cells can adapt to many stresses. They develop resistance to chemotherapy. They find workarounds when you block one metabolic pathway. But when you combine nutrient restriction with bacterial metabolites that directly induce cell death, you're attacking from two angles simultaneously. The cancer cells can't compensate fast enough. The bacterial strains that produce these protective compounds need specific foods to thrive. Fiber is the foundation, not the fiber in processed cereal or supplements. Real fiber from whole plant foods. Your bacteria ferment this fiber and produce compounds called short-chain fatty acids. Butyrate is the most important one for colon health. Butyrate does several things. It's the preferred fuel source for the cells lining your colon, keeping them healthy and resilient. It reduces inflammation throughout the digestive tract, and critically, it strengthens the intestinal barrier, preventing cancer cells from accessing nutrients that leak through a damaged gut wall. Studies measuring butyrate levels in people with and without colon cancer show a clear pattern. Higher butyrate correlates with lower cancer risk and better outcomes in those already diagnosed. The difference isn't subtle. People in the highest quartile of butyrate production have roughly half the colon cancer incidence of those in the lowest quartile. You can't buy butyrate in a bottle and expect the same effect. Your body needs the bacteria to produce it locally in your colon where it acts. That requires feeding those bacteria daily with the right substrates. Resistant starch is particularly effective. This is starch that resists digestion in your small intestine and reaches your colon intact where bacteria ferment it. Cooked and cooled potatoes, rice, and legumes contain high amounts. Green bananas are another excellent source. The cooling process after cooking changes the starch structure, making it resistant to your digestive enzymes but perfect food for your colonic bacteria. A clinical trial in Australia tested this directly. Researchers gave one group of participants resistant starch supplements equivalent to eating 2 cooked and cooled potatoes daily. After 8 weeks, they measured butyrate production and inflammatory markers. Butyrate levels nearly doubled. Inflammatory markers associated with colon cancer risk dropped by 30 to 40%. The control group eating typical starch showed no change. Polyphenols are the second critical component. These are plant compounds that give foods their deep colors: berries, dark chocolate, green tea, red wine. Your gut bacteria metabolize polyphenols into compounds that have direct anti-cancer effects. One polyphenol called ellagic acid, found concentrated in pomegranates and berries, gets converted by gut bacteria into urolithin A. Urolithin A activates a cellular cleanup process called mitophagy, where damaged mitochondria get removed and recycled. Cancer cells typically have dysfunctional mitochondria. Enhancing mitophagy selectively damages cancer cells while improving healthy cell function. Research from Switzerland showed that people who naturally produce high levels of urolithin A have significantly better muscle function, metabolic health, and lower inflammation markers. But here's the catch. Only about 40% of people have the right gut bacteria to convert ellagic acid into urolithin A efficiently. The rest need to build those bacterial populations through consistent polyphenol intake over weeks or months. Cruciferous vegetables add another layer. Broccoli, Brussels sprouts, cabbage, kale. These contain compounds called glucosinolates that gut bacteria convert into isothiocyanates. These molecules activate detoxification pathways in your liver and colon cells, helping them neutralize carcinogens before they can damage DNA. A study following over 50,000 people for 10 years found that those eating cruciferous vegetables at least 3 times weekly had a 28% lower colon cancer incidence than those eating them less than once weekly. The protective effect was dose-dependent. More consumption meant more protection, up to about 5 servings weekly where the benefit plateaued. But consuming these foods randomly won't maximize their effect. Timing matters. Your gut bacteria have circadian rhythms just like you do. They're most metabolically active during specific windows. Feeding them the right substrates at the right times amplifies their protective compound production. The research suggests consuming your fiber-rich foods earlier in the day when bacterial fermentation rates are highest. Your polyphenol-rich foods work best in the afternoon and evening. This timing synchronizes with your body's natural inflammatory cycles, suppressing inflammation when it tends to peak. There's one more bacterial angle that's critical. Some gut bacteria actually feed cancer cells. Certain strains produce enzymes that break down complex carbohydrates into simple sugars, even when you're restricting dietary carbohydrates. They're undermining your metabolic intervention from the inside. The bacteria responsible are typically overgrown in people eating high amounts of processed foods and added sugars. They thrive in that environment. When you suddenly restrict carbohydrates, these bacteria don't disappear immediately. They continue producing glucose internally, providing cancer cells a fuel source that bypasses your dietary restrictions. Eliminating these problematic bacterial populations requires a strategic approach. You can't just stop feeding them and expect them to vanish. They're resilient. You need to actively displace them with beneficial strains while simultaneously restricting their preferred foods. This process takes 4 to 6 weeks of consistent dietary intervention before the bacterial ecosystem shifts meaningfully. The practical protocol needs to address all these mechanisms simultaneously: metabolic restriction, bacterial optimization, and targeted compound delivery. That's where the exact food list becomes critical. Here's the 5-food protocol that addresses every mechanism we've discussed. Not 5 random superfoods. 5 specific choices that work synergistically to starve cancer cells while optimizing your bacterial ecosystem. Food 1 is raw sauerkraut. Not the shelf-stable kind sitting in vinegar on store shelves. That's dead bacteria and brine. You need refrigerated, unpasteurized sauerkraut containing live Lactobacillus plantarum and Lactobacillus brevis. These specific strains produce compounds that directly inhibit colon cancer cell growth while displacing the sugar-loving bacteria, undermining your metabolic efforts. A study from Poland analyzed 27 different sauerkraut samples for their anti-cancer activity. The ones fermented longest, between 4 and 6 weeks, showed the strongest cancer cell growth inhibition. The researchers identified specific metabolites produced during fermentation that triggered apoptosis in colon cancer cells while leaving healthy colonocytes completely unaffected. The dose that matters is half a cup daily, consumed with your largest meal. The living bacteria need food to survive your stomach acid. Pairing them with a meal buffers the pH and dramatically increases bacterial survival to your colon. Food 2 is green kiwi fruit, not golden kiwi, the green variety specifically. Green kiwis contain an enzyme called actinidin that breaks down proteins in your digestive tract, but that's not why they're on this list. They contain a unique combination of fiber types that selectively feed Akkermansia mucinifila, the guardian bacterium protecting your colon lining. Research from New Zealand demonstrated that consuming 2 green kiwis daily for 4 weeks increased Akkermansia populations by 370% in adults over 50. That's not a modest shift. That's a complete ecosystem transformation. Akkermansia produces a mucus layer that physically separates your colon cells from potential carcinogens while generating butyrate that directly activates tumor suppressor genes. Eat both kiwis in the morning on an empty stomach. The actinidin enzyme works best in a fasted state, and the fiber reaches your colon by afternoon when bacterial fermentation activity peaks. Food 3 requires explanation because most people have been taught the opposite. White rice. Specifically, white rice that's been cooked, cooled completely, then reheated. This process creates resistant starch type 3, a carbohydrate your body cannot digest, but your bacteria ferment into butyrate at rates exceeding any other food source. Japanese researchers compared colon cancer rates across populations consuming different rice preparations. Those eating cooled and reheated white rice had 41% lower colon cancer incidence than those eating freshly cooked rice, even when total rice consumption was identical. The resistant starch made the difference. Cook your rice normally, spread it on a sheet pan, refrigerate overnight, then reheat the next day. 1 cup of prepared resistant starch rice 3 times weekly provides your bacteria the raw material for massive butyrate production without spiking blood glucose. Here's why this matters more than you think. Butyrate does something remarkable beyond feeding your colon cells. It modifies the expression of over 200 genes related to cellular metabolism, inflammation, and cancer development. It's not just food for bacteria, it's a signaling molecule that reprograms how your colon cells function at the genetic level. Food 4 is raw cacao nibs. Not chocolate bars, not cocoa powder. The unprocessed cacao bean fragments that taste bitter and slightly unpleasant. They contain the highest concentration of polyphenols found in any food, specifically epicatechin and procyanidins that gut bacteria convert into metabolites with direct anti-angiogenic effects. Blood vessels feeding tumors require growth signals to maintain themselves. These polyphenol metabolites block those signals. A tumor with compromised blood supply cannot grow regardless of available glucose. You're implementing a dual starvation strategy, metabolic restriction plus vascular restriction. The effective dose based on bioavailability studies is 20 grams daily, roughly 2 tablespoons. Blend them into smoothies or eat them with a small amount of coconut oil to improve absorption. The bitterness tells you they're working. Processed chocolate loses most of these compounds during manufacturing. Food 5 is pressure-cooked lentils. The pressure cooking matters because it breaks down anti-nutrients that normally block mineral absorption while preserving the specific fiber structure that feeds Faecalibacterium prausnitzii, another keystone bacterial species for colon health. French researchers discovered that Faecalibacterium populations were depleted in 93% of colon cancer patients compared to healthy controls. Restoring this species through targeted fiber intake reduced inflammatory markers associated with cancer progression by 67% over 12 weeks. 1 cup of pressure-cooked lentils 4 times weekly provides enough substrate to rebuild Faecalibacterium populations even in depleted microbiomes. Add them to salads, soups, or eat them plain with olive oil and sea salt. Now, the timing protocol that makes these 5 foods work together instead of just coexisting in your gut. Morning first thing, 2 green kiwis on an empty stomach. Wait 30 minutes. This gives the actinidin time to work and the fiber time to begin its journey without interference. With your first meal, half a cup of raw sauerkraut. The living bacteria ride the meal through your stomach, surviving to colonize your colon. This is your bacterial reinforcement arriving when your system is most receptive. Lunch includes your resistant starch rice if it's a rice day. The afternoon fermentation window captures maximum butyrate production when your colon cells are most metabolically active. Afternoon, between meals, 20 grams of raw cacao nibs. The empty stomach allows faster absorption of polyphenols, maximizing their conversion to anti-angiogenic metabolites. Dinner, if it's a lentil day, include your cup of pressure-cooked lentils. Evening fiber consumption supports overnight bacterial fermentation, producing metabolites that regulate your morning inflammatory state. This protocol addresses three distinct mechanisms simultaneously. You're starving cancer cells metabolically. You're optimizing bacterial populations that produce anti-cancer compounds. You're delivering targeted molecules that cut off tumor blood supply. Most importantly, you're doing this through food combinations that amplify each other's effects rather than working independently. The sauerkraut bacteria help digest the kiwi fiber more efficiently. The resistant starch feeds bacteria that enhance polyphenol metabolism. The lentil fiber supports bacteria that improve your gut barrier, making all other interventions more effective. But there's one variable we haven't addressed yet that determines whether this entire protocol succeeds or fails completely. Something happening in your body right now that's either supporting or sabotaging every intervention we've discussed. That variable is inflammation. Specifically, systemic chronic inflammation measured through high-sensitivity C-reactive protein levels above 3 milligrams per liter. Because inflammation doesn't just interfere with one pathway in your cancer prevention strategy, it disrupts all of them simultaneously. When inflammatory cytokines flood your bloodstream, they degrade your gut barrier integrity, allowing bacterial endotoxins to leak into circulation. Those endotoxins trigger more inflammation in a self-perpetuating cycle. Your carefully cultivated bacterial populations can't colonize effectively in an inflamed environment. The polyphenols you're consuming get consumed fighting oxidative stress instead of blocking angiogenesis. The butyrate your bacteria produce gets diverted toward repairing inflammatory damage rather than signaling colon cells to remain differentiated and healthy. Research from Massachusetts General Hospital tracked inflammatory markers in 1,200 adults implementing gut-focused cancer prevention protocols. Participants with baseline CRP above 3 showed 62% reduced benefit from dietary interventions compared to those with CRP below 1. Same foods, same bacterial strains, dramatically different outcomes based purely on inflammatory status. You can eat every cancer-fighting food available, but if your body is burning with chronic inflammation, you're trying to fill a bucket with holes in the bottom. So before this protocol delivers maximum effect, you need to address inflammation directly. Not through anti-inflammatory medications that create their own complications, but through targeted lifestyle modifications that most people overlook completely. Sleep fragmentation is the silent inflammation amplifier nobody discusses. Not total sleep duration, but the number of times you partially wake during the night without remembering. Each micro-awakening triggers a cortisol spike that activates inflammatory pathways. Studies using continuous sleep monitoring show that adults averaging more than 15 micro-awakenings per night maintain CRP levels 70% higher than those with fewer than 5, regardless of total sleep time. The solution isn't sleeping longer, it's sleeping more continuously. 2 interventions backed by sleep research dramatically reduce fragmentation. First, complete darkness in your bedroom. Even small amounts of ambient light penetrating closed eyelids trigger partial arousals you won't consciously remember. Blackout curtains or a quality sleep mask eliminate this disruption. Second, consistent sleep timing within a 30-minute window every single night, including weekends. Your circadian system stabilizes when it can predict sleep onset accurately, reducing the neurochemical fluctuations that cause fragmentation. Implement both changes tonight. Within 2 weeks, your inflammatory markers will begin declining even before dietary interventions take full effect. The second inflammation driver is chronic psychological stress, but not the way most people understand it. Short-term acute stress actually enhances immune function temporarily. The problem is rumination, the mental loop where you replay past events or rehearse future scenarios repeatedly without resolution. Neuroscience research shows that rumination activates the same inflammatory pathways as physical infection. Your immune system cannot distinguish between a real bacterial threat and an imagined social threat you're replaying mentally. When subjects in controlled studies practice structured worry periods, setting aside 15 minutes daily to deliberately process concerns, then consciously redirecting attention afterward, their inflammatory markers drop by 38% over 8 weeks. Not meditation, not relaxation, just designated mental processing time that prevents all-day rumination. Schedule it, use it, move on. Third inflammation source is oral bacteria translocation from poor dental hygiene. The bacteria causing gum disease, particularly Porphyromonas gingivalis, don't stay in your mouth. They enter your bloodstream through inflamed gum tissue, triggering systemic inflammation that persists for hours after each exposure. Patients with untreated periodontal disease maintain CRP levels comparable to those with diagnosed inflammatory disorders. Brush twice daily, floss once daily, see your dentist every 6 months. Non-negotiable if you're serious about cancer prevention. Now, assuming you've addressed inflammation, here's the final piece that determines long-term success: This protocol isn't a 30-day experiment. It's a permanent dietary framework. The bacterial populations you're cultivating take 3 months to fully establish. The epigenetic changes from consistent butyrate exposure require 6 months to stabilize. The vascular remodeling that makes tumors unsustainable needs 9 months of sustained anti-angiogenic compound exposure. You cannot do this for 8 weeks, feel slightly better, then return to previous eating patterns expecting the benefits to persist. They won't. But here's what makes sustained implementation realistic. You're not eliminating foods you love, you're adding specific foods at specific times. Your regular meals continue. You're simply incorporating these 5 targeted additions into your existing routine. The green kiwis replace whatever you currently eat first thing in the morning. The sauerkraut sits alongside your regular breakfast. The resistant starch rice becomes your carbohydrate choice when you eat rice. The cacao nibs replace whatever afternoon snack you currently consume. The lentils substitute for another protein source at dinner a few times weekly. You're not overhauling your entire diet. You're making 5 strategic substitutions that compound their effects through synergistic bacterial and metabolic pathways. Most people can maintain that indefinitely because it doesn't feel like deprivation or restriction. Start tomorrow morning with 2 green kiwis on an empty stomach. That's the only change for week 1. Let your system adjust to that single modification before adding the next. Week 2, introduce the sauerkraut with breakfast. Week 3, swap regular rice for resistant starch rice twice that week. Week 4, add the cacao nibs. Week 5, incorporate lentils twice. By week 6, you're running the complete protocol without having shocked your system or overwhelmed your routine. Track one simple marker: your bowel movement consistency and frequency. When this protocol works, you'll notice changes within 10 days. More regular timing, more complete elimination, less bloating. That's your gut bacteria responding to their new fuel sources. That's butyrate production increasing. That's your colon cells receiving the signals they need to maintain healthy differentiation. That's cancer prevention happening in real time at the cellular level. Everything else you do in life matters less for your colon health than what happens in your gut microbiome every single day. Your colon cells are making a decision right now about whether to stay healthy or begin the mutations that lead to cancer. That decision depends entirely on the signals they're receiving from your bacterial ecosystem and the nutrients in your bloodstream. Those signals are under your direct control, starting with your next meal. Stage 4 colon cancer survival rates have jumped from 13% to 60% in a decade because researchers stopped trying to just kill cancer cells and started understanding how to starve them selectively. The metabolic vulnerabilities cancer cells have, their addiction to glucose and glutamine, their inability to use ketones, their dependence on angiogenesis. These aren't theoretical weaknesses. They're exploitable targets you can act on today. The bacterial strains producing butyrate, the resistant starch feeding Akkermansia, the polyphenols blocking tumor blood supply, none of this requires prescription access or specialist supervision. It requires consistency with 5 specific foods eaten at strategic times. 2 green kiwis first thing in the morning, half a cup of raw sauerkraut with breakfast, resistant starch rice 3 times weekly, 20 grams of raw cacao nibs in the afternoon, pressure-cooked lentils 4 times weekly. That's the protocol. That's what the research shows works at the cellular level to prevent and potentially reverse colon cancer development. You already know what happens if you ignore this information. The statistics are clear. 1 in 23 people will develop colon cancer in their lifetime. Those odds drop dramatically when you implement these targeted interventions. Your gut bacteria are waiting for instructions. Your colon cells are responding to whatever signals they're receiving right now. Give them the right signals consistently, and cancer cells lose the metabolic environment they need to survive. It's that direct.